All right, so I think I'll start by just giving some background on myself and how I got into this area of research. Um, so going back, so I'm from Kansas, and I did a science fair project every year while I was in elementary school on a different topic. And when I was in sixth grade, I was trying to decide what topic I should do that year. So my best friend at the time, she had fallen off a horse and she hit her head on a concrete block. And then following that, she started to have seizures. So I thought that that year I could look into epilepsy as my topic for that project. So I contacted the epilepsy group at the University of Kansas Medical Center, got in touch with them. They invited me to come visit. Then they actually said that I could watch while they did a surgery where they implanted electrode contacts directly on a patient's brain. And they showed me how they collect intracranial EEG data from those epilepsy patients. So I saw how they did it for that particular patient. And then they showed me how to visually inspect that data and then how to look for seizures or other epileptiform activity in the data. So that's how I started working on epilepsy research. And then with that group, I continued working with them for a summer in high school. So I did a research experience for undergraduates while I was in high school. And I did research with them doing EEG data analysis. And then when I went to college, I went to the University of Chicago and I studied math over there. So it was mainly pure math, and I thought that that was a good foundation for the applied work that I wanted to do later on. But besides that, so I did a double major with Polish literature and a minor in computational neuroscience. And then after that, I went to Yale and I did a PhD in electrical engineering, but my advisor there was from mathematics. And then I had a co-advisor from neurology. So that's when I got back into epilepsy research. So it was more about developing analytic tools and different types of methods to analyze intracranial EEG data. And I was trying to predict seizures in epilepsy patients, but also study different types of networks in the brain. So one type of network that I was studying is called a resting state network. So we were trying to see if, based on the work that had been done on resting state networks using fMRI, if we could also see the same types of networks in epilepsy patients looking at their EEG data. And then following that, once I graduated, I went to Stanford and I did a postdoc in the neurology department over there. So I worked at the med school and I worked with epilepsy patients. I made experiments. So I developed the experiments and gave them to the patients while they were, while they had the intracranial electrodes on their brains collecting data. And then after that, I did a short postdoc at UC Davis in the math department, and then finally ended up where I am now at the Neuroimaging and Informatics Institute. So what I really like about our institute is that it's very multidisciplinary. So my background is mixed. It's engineering, math, and neurology. And I feel like all of those areas come together at our institute, and it's very collaborative. So that's where I am. I've been here for the past 5 years and mainly focusing on epilepsy following traumatic brain injury. So I'll give you just a little overview, some background on epilepsy if you're not familiar. So epilepsy is a neurological disorder that affects the nervous system, and it's exhibited by seizures. So if someone has at least 2 unprovoked seizures, that means that they have epilepsy. And the cause is usually unknown, which makes it really difficult to study. But for cases in which we do know the cause, it could be from brain damage, congenital abnormalities or genetic conditions, head injury, stroke, infection of the brain, genetic syndromes, or brain tumors. And a seizure is a sudden surge of electrical activity in the brain. This is why we tend to focus on looking at EEG data when we study epilepsy, because that measures the electrical activity in the brain. And seizures may last for a few seconds or a few minutes. They could be frequent. A patient might have hundreds of seizures per day. They might just have them once every few years. So there's a lot of variability there. And seizures take various forms. So I think people often think that seizures are the type where a person is convulsing, but there are different types of seizures, like absence seizures, in which a patient loses consciousness, but it looks like the person just kind of is staring blankly. So that's also a form of seizure. And some people sense an aura before their seizures occur. So it's some type of warning, and this varies depending on the person. So sometimes they see something, sometimes they hear something, sometimes they have a weird smell that occurs. They might have numbness or tingling or other feelings that are associated with something that occurs right before they have a seizure. And 36% of people do not respond to medications. So it's really tricky to control seizures. In terms of numbers, it's a big problem. 1 in 26 Americans will develop epilepsy in their lifetime. 3 million Americans currently have epilepsy, 65 million people worldwide. And going back to medication, so I said that 36% of people cannot be treated with medications, but 47% are seizure-free with one drug. Other people have to have several medications, and then that is able to treat their epilepsy. So another way that we can treat epilepsy is with the ketogenic diet, which I find interesting. So many of you might know about this diet. It's high fat and low carb. And it's difficult to get people to be on this in a very strict way. And it has to be strict for it to work. But it's not really known why it works, but for whatever reason, the ketogenic diet tends to work in some people in stopping their seizures. Other ways to treat epilepsy is with implantable devices. So there's something called the NeuroPace, which looks like this here on the bottom right. And it's a little device. So backing up, if someone develops seizures and they have epilepsy, typically they'll have scalp EEG, and the neurologist will be able to look at that. And based on the scalp EEG that are placed on the surface of the head, they can see if they think that one particular part of the brain is causing the seizures and it's localized to one area, or it might be a network, it might be a large part of the brain that is causing the seizures. But if it's localized, then they might do a surgery where they open up the person's head, they have access to the person's brain, and then they implant those intracranial electrodes on the person's brain. And then they can get recordings of the electrical activity all over the brain and deep within the brain. So based on that, they can determine if the seizures are actually localized to one part of the brain. And if a person has epilepsy that's localized, they have a couple different options. They can have another surgery where the neurosurgeon resects that part of the brain. So actually cuts out and removes that little part of the brain that is causing the seizures. And that might result in other side effects, obviously depending on what part of the brain they're cutting out. But it tends to be very effective in stopping the seizures from occurring. But going back to these implantable devices, if the epilepsy is localized to one part of the brain, they can cut out part of the skull above that part of the brain and replace it by this little device. And this little device has 2 electrode contacts. One of those records data from that part of the brain, and the other one will provide electrical stimulation whenever it decides the person is about to have a seizure, to try to counteract that seizure activity and stop it from happening. So what it does is it tries to learn a particular patient's pre-seizure activity. So the algorithm improves over time with more data that are collected. And for this device, they were claiming 50% reduction in seizures in about 50% of patients. So pretty good results. Yeah. So intracranial EEG, this is what it looks like. You might have grid electrodes or strip electrodes or depth electrodes. This is a picture of what they look like. So the strip electrode here on the bottom, you just place them wherever you want to get recordings. So you might place that grid, that 8x8 on top of the brain, and then you might rearrange strip electrodes around that. This depth electrode goes deep within the brain, and it has electrode contacts along that electrode, and each of those contacts records the electrical activity from that particular part of the brain. So this is what it would look like. So from one of those electrode contacts, we get this type of time series data. So we get the electrical activity over time, and it's continuous recordings, sometimes 256 samples per second, sometimes more, sometimes a couple thousand samples per second. And if you have many of these electrode contacts, you can imagine you end up with very large matrices of data, because you might be recording data for 1 week at a time. And if it's a couple thousand samples per second, you have a very large matrix that way, and it might be 20, it might be, if you're doing intracranial EEG, it might be a couple hundred electrode contacts. So then one of the questions in terms of analysis is, how can we extract the most important underlying brain activity from those really large matrices, so that we can reduce the dimensionality of those large matrices, and then try to identify patterns in the data that can help us with either seizure prediction or whatever problem we're interested in. So another thing that's very important in what we study is neuroimaging. So we look at CT scans, MRI, DTI, fMRI, PET scans, SPECT studies, and MEG. So I'll go into these in a little bit more. But for one instance is for structural MRI, sometimes we see changes in the structural images of the brain after seizures. So here we can see some differences. We can see some local swelling in the brain. And so going back to what I said about how most of the time the cause of epilepsy is unknown, one of the causes of epilepsy that we do know about is traumatic brain injuries. And so that's why we decided for this study to focus on that, because we know that a certain percentage of people will develop epilepsy after a traumatic brain injury. So we just wanted to study that. So in terms of traumatic brain injury, there are different classifications. We have mild, moderate, and severe TBI. And so mild TBI would be like a concussion. There are no skull fractures, and the loss of consciousness that the person experiences would be less than 30 minutes. And then the more severe the injury is, it would have a more severe classification. And the more severe the injury is, the more likely the person is to develop epilepsy later on. So what is post-traumatic epilepsy? It is recurrent seizures following a traumatic brain injury. And let me skip over that. So this is the timeline of the progression of post-traumatic epilepsy. So a person would have an initial traumatic brain injury, and then they might have immediate seizures within the first 24 hours, but that doesn't mean that they have post-traumatic epilepsy. Then in that first week, they might have early post-traumatic seizures, but it's not until at least 1 week after the injury that people may have late post-traumatic seizures. And then if someone has at least one of those, they would be diagnosed with post-traumatic epilepsy. But it's tricky to study this because sometimes people don't develop those late post-traumatic seizures until many years after having a traumatic brain injury. So it might be more than 10 years. So the challenges of studying post-traumatic epilepsy are that the majority of subjects who suffer TBI never develop epilepsy. It's often many years before seizure activity is recorded, and each of the stages of post-traumatic epilepsy may be associated with different biomarkers that we're studying. So I'll go into that in a little bit now. So a biomarker is an unambiguous biological characteristic of normal or pathological processes. So for the case of epileptogenesis, or the development of epilepsy, there are quantitative measurements that can be used to identify any kind of epileptogenic abnormalities that could predict the development of epilepsy. So what we want to do is we want to look at different data types right after the traumatic brain injury, and based on that, can we find any patterns, any kind of indication from that data that can tell us who is most likely to develop epilepsy later on. And then that can help us in terms of treatment and giving medications and different types of therapies for those people who are likely to have it. So biomarkers can take numerous forms. So they can be from biological imaging or electrophysiological data. And here's another list of potential biomarkers, but we're just going to focus on EEG, imaging, and blood data. So several key imaging biomarkers have been found to distinguish epileptic from non-epileptic patients. So for MRI, some people have found that the lesion severity and MRI abnormalities like signal changes could be biomarkers. Different types of morphometry in the MRI, like looking at the volume of different structures in the brain, such as thalamic and hippocampal volume. And in fMRI, I'll skip over the PET stuff, but in fMRI, sometimes we see a reduction in functional connectivity, especially cortical thalamic projections. And in DTI, sometimes we see alterations in fractional anisotropy and mean diffusivity. So we started this big study called EpiBios for RX, and we have 7 principal investigators from all over the world. So it's from USC, UCLA, Melbourne, University of Eastern Finland, and Albert Einstein College of Medicine in New York. And we're looking for treatments and associated biomarkers of epileptogenesis. So the challenge is that this process is likely multifactorial and crosses multiple modalities. So that's why we're looking at different types of data. So let me give you an overview of this study. So we're doing a parallel study looking at humans and rats. So we're collecting similar types of data for both of them. So for the TBI patients, we are going to look at 300 patients, and they're coming in on a rolling basis. So right now we're at about 175 people enrolled, and we're collecting longitudinal data. So we follow them for 2 years, and then we collect data at different time points throughout those 2 years. And we collect EEG, imaging, and blood data. And then, so similarly for the rats, except for the human patients, they all have had a moderate to severe traumatic brain injury, whereas with the rats, the injuries are induced, and so we can control and make sure that the level of severity is the same across all of the rodents and the location of the injury. And you see this little hammer here. It's not exactly like that, but it's on a pendulum and it swings, and it hits them in the head and causes the injury. But we don't do any of the experiments at our place. Different sites do the experimental work, also collecting data from the human patients, and then they send us, so Arthur Toga and me at USC, they send us all of their data. We have developed a data repository. We help in organizing the data, managing it, storing it on our server, and then also with sharing it to the broader epilepsy research community. And in addition to that, we're also working on developing analytic tools. So we want to develop methods in which we can analyze different types of data, such as EEG, imaging, and blood data, and also assist all of these investigators that are working on this project to try to expedite the research and identify biomarkers of epileptogenesis after a TBI. So once we identify those biomarkers, which will likely be this coming year, then our collaborators are going to test out 5 different medications, the ones listed here in gray, and they'll use those on rodents in a preclinical trial. So based on the biomarkers that we identify, they'll know which rodents we think are likely to develop epilepsy, and we'll have different cohorts of rats, and they'll be given these different medications, and we'll see which of them or how many of them actually develop epilepsy. And based on the outcome of that preclinical trial, we'll hopefully start to prepare for a clinical trial in humans. So we have developed something called the LONI pipeline. So LONI for the Laboratory of Neuroimaging. And this is a free tool, so if you search for it, you can download it. And we have organized a library of different types of analytic tools written in different languages. So in R, in Python, in MATLAB, all kinds of different programming languages, but we have them organized in this library in one place on this one tool. And we also link this with our data repository. So you can organize the analysis that you want to do. You can pick out these little modules and then connect them to each other, and then upload the data that you're interested in analyzing it, and then it'll go through all of those steps, and then you'll get your outcome. So one thing that we do is lesion mapping, because we're very interested in seeing the location of the lesions for the patients, because there's so much variability. Like I said, unlike the rodents that have the lesions in one place, with the humans, the lesions are located all over their brains. And so we need to identify where those are, and then see if there's some relationship between either the severity of the lesion or the location of the lesion and which patients end up developing seizures. So here we superimposed the lesions on one brain, but this is about a dozen patients and their lesions. So, so that's just one more. I'll skip ahead. So we're also doing work with the diffusion MRI data. So for the rodents, I'll show this was kind of an interesting result that we found. If we compare 45 sham rodents with 45 rodents that had a traumatic brain injury, and we averaged across those 45 in each group, we can see that there's a loss of connectivity in the rats that had the injury. And then looking at it differently here, looking at the corpus callosum connectivity, we see down here at the bottom, there was the lesion, that's where it was located. And so we see a loss of connectivity there, but then we also see a loss of connectivity on the contralateral side up here. So that was pretty interesting. And then we do similar work with the humans with the diffusion MRI. And then going back to the EEG data, one thing that we are looking at as a potential biomarker of epileptogenesis are high frequency oscillations. So this is a type of pattern that we often see. We often see more of these patterns before someone starts to develop seizures. And we have this Ripple Lab toolbox in MATLAB that automatically detects these HFOs, but it often ends up coming out with lots of false HFOs in addition to the true HFOs. So it'll identify many artifacts and noise as HFOs. So then we need to manually and visually inspect all of those events that were identified and determine which ones are true HFOs and which are not. So, so the patterns that we look for in these HFOs, we want to see at least 4 oscillations. We want to see that the amplitude of the HFO, that it's at least twice as big as the amplitude of the EEG before and after. And then also we look down here at the time frequency plots, we look for a circular pattern that's yellow in a certain frequency range. So over here on the right, we see an example of something that was identified as being an HFO, but it was actually a muscle artifact. So down here we see these columns, and then we see this repetitive pattern here in the EEG, and this is something that we see typically in the rats because they're moving around. So the EEG are also very noisy, and so one thing that we need to do is we need to develop filtering methods to extract the noise and filter the data accurately. So we do stuff like notch filtering and surface Laplacians and some other methods to try to remove the noise. And then with those HFOs, besides just identifying them, besides counting how many each person has, we're also interested in seeing which part of the brain is generating most of these HFOs. So here on the left, we can see for this patient, most of them were located down here in the red area. And then what we can do is we can relate that to the imaging. So we can see what was happening with the imaging, where were the lesions located, is there any type of correlation between the location of the lesion and the location of where most of these HFOs are occurring, from which electrode contacts. And with the lesion segmentation, we have some automatic tools that do this for us. And sometimes it's done accurately, like you see here. So you can see in the red, that's where the automatic tool was able to identify the lesion accurately. But in other cases, because it's a pretty complicated problem with these TBI lesions. So sometimes it messes up and it doesn't accurately identify the lesion. So in this case here on the right, it identified the surrounding edema, but not the lesion itself. So I have some students in my lab that are doing manual segmentation of these lesions, and then we're working on collecting enough of those manual segmentations so that we can work on developing more accurate automatic segmentation methods, so we can train a system to do this automatically. So I just wanted to, I didn't want to spend too much time on this, but I wanted to talk about another tool that we developed that helps with this segmentation of MRI data. And we decided to do this using, let me just skip ahead here. So FreeSurfer is one of these software types that we use for the segmentation. And it's about 80 to 90% accurate. So what we do is we have it run automatically, and then students go into the FreeView interface of the FreeSurfer software, and they have to go slice by slice, manually correcting any errors that were made. So we decided to develop a tool using virtual reality, thinking that this might be a more intuitive way to go about this problem of correcting segmentation errors. And it might be more efficient because we can view the three-dimensional data three-dimensionally instead of being limited to one slice at a time. So I'll show you, I'll skip ahead. I'll show you this little video without music. So this is, you can see one of our programmers, Adam, is looking at MRI, and that yellow outline was generated by FreeSurfer. So the yellow outline here is separating brain tissue from non-brain tissue, but it messed up around the optic nerve over here. So he's going to zoom in using the HTC Vive. He's going to zoom in on just the optic nerve region, and he's going to use one of those controllers, and anytime that red sphere intersects with any of those voxels, he can delete them. So his goal here is to delete those voxels in that yellow outline that was mistakenly put there. And what he's going to do is then save that and then run that through FreeSurfer one more time to force it to make that correction so that it won't make that yellow outline in that area where it shouldn't be there. So he can see multiple slices at a time. We created spacing in between those voxels so that you can be very precise about which ones you are deleting. And you can see that the little boxes in front are a bit smaller than the ones behind so that you can see what's back there. And you can scroll through the layers with one of those controllers very easily. So, and we did user testing on non-experts, and we trained them to do this on the computer using FreeSurfer, and then also using the virtual brain segmenter. And we got promising results, and they were able to complete their task faster and more accurately using the virtual brain segmenter. And so we're hoping that this will be a way for us to crowdsource some of these issues and then use some machine learning to improve the segmentation, the automatic segmentation. So yeah, so we're going to gamify this later on and then crowdsource it and hopefully collect a lot of data on it. And I just briefly wanted to talk about some of the other work that we're doing in terms of data repositories. So we started a project about 2 years ago called the Data Archive for the Brain Initiative. And so we've developed a data repository for projects that collect human invasive recordings for different brain initiative projects. So some of them are on epilepsy, others on Parkinson's or depression. And we've developed this nice repository for people to use where they can request access to data, and then other data are publicly available. And we've also linked that repository with other quality control tools, visualization tools, so they can visualize the imaging as well as the EEG data, and also different types of statistical and analytic tools. So it's a way for people to not have to download data onto their local computers and then run their own analyses, but they can run analyses directly on our server through the web interface and share their findings with the broader research community. So based on that work, I wrote a proposal in March to do something similar on COVID-19 data. So this project started in May, and we have also developed a data repository. Right now we're focusing on chest CT images, but we're also including other types of data related to COVID-19 patients, and also including similar quality control, visualization, and analytic tools for people to use. And so we're actively working on this, and also working on using some deep learning to distinguish between those who have COVID-19 and those who don't, based on chest CT images. So I'll briefly show this video. Does it not want to play? Okay, so just kidding, but you can see it on the website if you go. And so this is just an example of some of those chest CT images. We have images from COVID patients and images from non-COVID patients. And we're using different types of automatic tools to distinguish those two groups with a pretty high accuracy. And I just wanted to include some brain art from Keck from USC. And I wanted to leave time for discussion and for questions. And so I'll finish there, and I'll see if, I don't know if I had any chat questions. Here's the chat. No, so nothing yet. Okay, but please ask me any questions. Let me know what you want to hear more about. And yeah. Let's open it up for questions now.
Oh, thanks, Dr. Duncan. That was definitely super informative. So while we were waiting for others to send their questions in, I personally had one because I was working on CT scans or diagnosing COVID-19 from CT scans a few months ago. And I was just wondering what metrics you use for verifying your accuracy, because our team was able to attain around 98, 99% accuracy, but we weren't sure as to how you analyze whether you're overfitting or just not having enough diversity in your data sets.
Yeah, so we're working with neuroradiologists and radiologists. And so we're also like right at that point right now. So, so it's a work in progress, but that's definitely something that we're concerned about too.
Something a little similar to the question Ox just asked. Himita just asked, what kind of machine learning tools did you use to differentiate between COVID and non-COVID CTs?
So we have all kinds of, here, let me pull up a document. It's up here. Yeah. Okay. So these are the data sets that we're using. And then like all the students on my team are working on different types of methods here. So some deep learning, some CNN models. So one person is using ResNet to extract features. What else? There was one study that did a comparison of different methods, but also looking at different data. So we wanted to try to reproduce that, but using one data set. Yeah, there's something, someone's looking into dark COVIDNet. That's something that they found. COVNet. Coronet. So there are many, but we're working on showing some of these preliminary results and sharing some of the methods that we felt worked pretty well on the website. So we'll probably add those in the next month or so, and also some transfer learning. So yeah, so stay tuned on that, and we'll be able to say more coming up. How dangerous are the various types of electrodes you place on the brain with respect to others? They, like in terms of causing infection or something like that, like that is a risk, but they're generally very safe, and they're well tested. Yeah. Coming from a pure math background, what caught your interest in computational neuroscience? So my parents are math professors, and they definitely heavily pushed math on me at an early age. So I started taking math classes at the University of Kansas when I was 12 years old. And then by the time I was in high school, I was taking some graduate level math courses at KU. So I definitely felt like having a strong foundation in math was very helpful for whatever I wanted to do later on. But I was always interested in working on the brain, and I thought that there are many interesting problems. Like I worked on Alzheimer's for a couple years, and that was very interesting as well. So yeah, I added that minor in computational neuroscience my senior year of college, because I just wanted to get more into that and start to move away from pure math into more applied math and engineering work. So that's why I felt like in grad school I did mainly applied math, even though my degree was in electrical engineering. And then, yeah, that's kind of how I got into it. Yeah, let me pull up this one. So, so let me show you some slides about what we do with the dimensionality reduction. So let me close this for a second. So, so we want to study the variability of the statistics of the EEG. We have these really, really large matrices, and some of the things that we consider are covariance matrices, local power spectra, something called the Mahalanobis distance between local covariance matrices, and we want to see how these statistics change over time, and then can we use these methods to distinguish between those TBI patients who develop epilepsy and those who don't. So by taking a manifold learning approach, I don't know how much math most of you have had, but this is a way that we can reduce the number of features while retaining the maximum amount of information from our EEG data. So we compute a kernel with a specially tailored distance measure, and then we employ various types of manifold learning techniques to recover the underlying brain activity. So one of these is diffusion maps. And if any of you have had linear algebra, you may have learned about eigenvalues and eigenvectors. So here, what we do is we take eigenfunctions of Markov matrices, and these are used to construct coordinates that generate efficient representations of such complex geometric structures like the brain data that we're dealing with. So diffusion mapping allows us to reduce the dimensionality of our large data sets and also identify patterns in the data so that we can distinguish between the two groups, those who develop epilepsy and those who don't. So this is a method that can be compared to principal components analysis, if any of you have used that. But diffusion maps are nonlinear and local, so it tends to be a little bit more applicable to complex brain data. So just skipping ahead, so this is kind of what it looks like. This is an old result, but I was looking at using diffusion maps to predict seizures or to identify pre-seizure activity in patients. And each of these dots in this embedding represents a window of data. And what we do is we start with high dimensional data, and then we embed that data using eigenvectors and corresponding eigenvalues, and we bring that into a smaller, for example, three-dimensional space. And then we can plot these new points in that three-dimensional embedding, and we can see that most of them are scattered in a cloud, sort of around the origin here, but we have some outliers, and the color here represents time. So time leading up to a seizure, but not including the seizure. So we saw that those red points that are close to where the seizure begins, they start to move away from the origin. So those outliers can indicate that a seizure might be coming. So then what we can do, besides just visually inspecting that embedding, we can take each of these points in the embedding and take the Euclidean distance to the origin of each of those points and plot a new graph with those distances. And then we can see that leading up to the seizure, there's a lot more variability here because those orange and red points are kind of bouncing back and forth between the origin and being outliers. And then I developed something called unsupervised diffusion component analysis initially when I worked on Alzheimer's, but then I applied this to, and that was with MRI data, but then I applied this method to post-traumatic epilepsy using EEG. So, it's a similar concept though. And so we're looking for these outliers in the embeddings and seeing if that can indicate that either a seizure is coming or if that can be a better indication of spikes or other epileptiform activity in the brain. So we did this for spike detection also, and looking at epileptiform abnormalities in the brain. So. Could you elaborate a little more on your Alzheimer's research? So yeah, so basically what I just said, I developed this, this method when I worked on Alzheimer's. And what I was trying to do is take structural MRI from mild cognitive impairment patients and trying to distinguish between those and healthy brains, so healthy MRI from healthy subjects, and seeing if using this method, if we then did the embeddings, if we could cluster out the healthy brains versus those with MCI, with mild cognitive impairment. So we were able to do that. What are some unique challenges with working with the brain now that we're working with designing interfaces? I mean, there are many, many challenges with working with the brain. It's just very complex, and there's a lot that we don't understand. And I feel like with epilepsy, for instance, sometimes the more we study it, the more we realize we know very little. So now, like, in terms of trying to cure epilepsy, the community has moved away from trying to cure it and focusing more on treating it. Does that kind of answer the question? Let me know if you.
That was good. Thank you.
Okay.